Hopefully you can see me there. Just once again, thanks Andrew, and good afternoon. I'll cover novel cannabis delivery, in particular a new delivery system that we at AIMC Biopharma's developed, which I'll cover towards the end of the talk. But before I start, I think it's good to give some background on cannabinoid pharmacology. So THC and CBD is the two main cannabinoids of interest at present, both highly lipophilic, and that's really important from the perspective of how those compounds can be absorbed into the body, which we'll cover a bit later. The molecular weights are 315 grams mole. Again, this is important when we're looking at, and I heard in the previous discussion about either transdermal delivery, absorption across the skin, or through mucous membranes, which I'll speak about later, where having a molecular size of less than 500 is what you really want to effectively absorb it across those membranes. Being highly lipophilic, it redistributes to, once it enters the body into the blood circulation, it redistributes very quickly to the highly perfused tissues like the liver, the lung, and the brain. And as mentioned earlier, it accumulates in the fatty tissues and then eventually will release back into the system and be eliminated. These compounds cross the blood-brain barrier. We know that they're psychoactive. Studies have shown that despite that, only less than 1% of THC actually of the dose administered reaches the brain. They readily cross the placenta and concentrate in the breast milk. And so even though there's a long usage history in pregnancy and even in breastfeeding over time, these are considered contraindications. The metabolism is primarily through the cytochrome P450 system in the liver through hydrolysis. And this is an important consideration for pharmacodynamic drug-drug interactions and pharmacokinetic drug-drug interactions, but particularly with the latter, with CBD, which also actually inhibits these enzymes and can increase the drug levels of other cannabinoids, as well as other medications that the patients may be on. And then these are further metabolized typically through phase 2 to glucuronides, which are more water soluble and can be excreted through the kidneys. So for THC, the primary metabolite is the 11-hydroxy THC, which is actually psychoactive, and it's formed very quickly after THC enters the blood circulation. And then there's the carboxy, which is considered non-psychoactive. Less is known about the CBD metabolites in terms of their pharmacology, but again, hydroxy and carboxy metabolites are seen in the blood shortly after. Looking at current delivery methods, so what's out there now, and just some of them would be, the main ones would be smoking and inhalation. These studies were done on the bioavailability through these routes many years ago, back even as far as the early 80s by Olson and his group. The THC there showed ranges between 10 and 37%, absolute bioavailability, CBD around the 30% mark, but highly variable. In terms of apparent and metabolite ratio, so there's more of the active drug than the metabolite when you're administering it in this route of administration, from 3 to 1 to 10 to 1, particularly when we're looking at 11-hydroxy THC to the hydroxy THC metabolite. Another study showed that the concentration of the drug declines very rapidly, 50% after 15 minutes of reaching its peak plasma concentration. And again, that's largely due to the lipophilicity of the drug. So the advantages of this method is obviously it's rapid drug delivery, fast onset of effects. The time to maximum concentration, Tmax, was 10 minutes in a number of studies or thereabouts. And the relatively high absorption of bioavailability. And also an ability to titrate to dose given the rapid onset of effects as well. But there are a number of disadvantages, and you know, when you're lighting anything on fire, it produces a lot of toxic compounds through smoking, pyrolysis, and combustion at the temperatures that the planter is exposed to. So aromatic hydrocarbons, carbon monoxide, ammonia, and so on. Vaporizing, however, uses lower temperatures, but there still are degrees of carbon monoxide and some very low levels of aromatic hydrocarbons that are formed as well, something to consider. And chronic usage is often associated with respiratory symptoms like bronchitis, cough, and sputum production. One of the biggest limitations is is highly variable in its absorption, and this can be related to smoking and the breathing dynamics that the user has at the time or from time to time. Also, the short duration of effect and the risk of passive exposure. We look at oral consumed or ingested products. The bioavailability is lower. We're looking at single digit or low double digit for both CBD and THC. When we look at the parent, there's a greater increase of the metabolites as the drug is now being metabolized through the liver. So the parent and metabolite ratios in the case of THC approach 1 or even in excess of 1, which is multifold higher than what you would see with the inhalation route of administration. And we do see intrahepatic circulation where the drug is reabsorbed and then excreted through the bile. And this can lead to seeing multiple peaks, or at least 2 peaks in a number of studies that have been published, and particularly with THC. The advantages are, of course, convenient and fixed dosing. The duration of effect is longer, and partly this is due to the active metabolites, particularly in the case of THC. On the flip side, though, there is slower and more variable absorption and lower bioavailability. If we look at oral mucosal, and I'll cover this with what we're doing in IX as well later, but the aim is to bypass first-pass metabolism. And that can occur if it's effectively absorbed across the mucosal membrane. Otherwise, it's invariably swallowed, and then you have the same issues which we have with an ingested product. Most of the research that's been done and published, at least on oral mucosal delivery, has been done with the oral mucosal spray Sativax, Nabiximols, and sublingual drops by GW Pharma. And so there is some data in the literature there. The advantages, again, it's convenient, it's more discreet than smoking or even vaporizing with the larger devices. And there's potential for faster absorption and onset of effect than oral ingested products. The disadvantages are slower absorption and lower bioavailability than the inhaled routes. And invariably, for most formulations, a high proportion of the active drug, of cannabinoids, are swallowed. Rectal absorption or rectal delivery through suppositories has been around for some time. This again aims to reduce first-pass metabolism, as the drug can be absorbed, bypass the portal vein in the liver and go directly into the systemic venous circulation. And some studies back from Renesa back in the 90s, when they used a hemisuccinate salt, so that they could double the oral bioavailability through the rectal route of administration. So there's the advantage of the bioavailability improvement over oral, but of course it's inconvenient and the lower compliance generally, as well as the slower and lower absorption profile compared to inhaled. And then transdermal, which I heard spoken about in the previous discussion. Again, with the high lipophilicity of the cannabinoids, it retards the diffusion across the aqueous layer of the skin. So permeation enhancers are often required, but if successful, this can bypass first-pass metabolism as well. And there's animal data to suggest that CBD is more permeable than THC. The advantages is it's convenient in chronic dosing situations. It can be quite useful, and particularly where compliance is an issue. For example, a patient with dementia, easier to apply a transdermal patch and leave it there in place for a longer period of time. The advantages of longer duration of action, but there are disadvantages. There is slower onset, slower offset too. So if you want to discontinue the medication, it's not easy to just stop it. Obviously, there are the cannabinoids, being fat soluble, accumulate in the fatty tissues and even the fatty tissue under the skin for a period of time. And there are variable durations of effects, typically with transdermal formulations, and tolerability. Local tolerability issues can be a problem, either with the excipients or the adhesives used. There is a transdermal gel in clinical trials for some orphan indications at present, but the pharmacokinetic data has not been made public, so it's hard to get a gauge on exactly the level of systemic absorption there. So there are a number of ways in which groups have tried to improve drug absorption. This is not specific to cannabinoids, but generally to looking at lipophilic or fat-soluble actives that are poorly absorbed. And liposomes is one way in which people have looked at this. They are microscopic vesicles So you've got a water component, a region, aqueous region that's surrounded by a lipid bilayer. And the drugs that are lipid soluble fit within that lipid bilayer. And these are very small. They're 30 nanometers to several micrometers. And these can improve the delivery of poorly water-soluble or fat-soluble cannabinoids. And they also can protect them from degradation like the gastric acid. There are limitations in terms of drug loading and how much you can put onto the liposome and stability in the circulation, and some manufacturing challenges with batch-to-batch consistencies and issues around scale-up. Micelles are a similar type of technology. They're similar vesicles and use often similar phospholipids or other surfactants, but contain a central hydrophobic or water-loving region, and fat-soluble loving region. And the advantages here, again, are very similar to the liposomes. Disadvantages, again, can be stability. And if you have other suspended solids, that can cause the CBD or cannabinoids to agglomerate. Nanoemulsions is another way to look at this. And this is the oil and water emulsions where the cannabis oil is stabilized by one or more dispersants. And typically, these are created by high-energy ultrasonic processes that disperse the oil phase into nanosized droplets, which can be as small as 25 nanometers. And the difference here is that they have the presence of a carrier oil as opposed to the micelle. And again, the same reasons to improve bioavailability. They can do so over microemulsions. They typically, though, require a high surfactant to active drug ratio, and that can limit the concentration in the final raw material drug product. And so therefore may not be suitable for certain finished products or for certain administration routes. One way or one subtype of nanoemulsions is what is called a self-emulsifying drug delivery system, which could be a self-gel capsule, contains a mixture of the active compound combined with lipids, surfactants, and a co-solvent. And when that reaches the aqueous phase of the gut, it actually breaks down and forms its own drug encapsulated oil water nanoemulsion. And these can be nanometer particle diameters as well. And the last one I'll probably just touch on is solid lipid nanoparticles. These are essentially emulsions where the liquid lipid phase has been replaced with a lipid that is solid at room temperature. And the advantages, body temperature rather, and the advantages of this, it can better host cannabinoids in this case and protect them from degradation and limit the diffusion of the actual cannabinoid or the active, whatever the case may be, to the outside surface of the particle. I'll move on to a novel delivery system that we've spent over a decade developing and now creating pharmaceutical products and now cannabinoid products at AIMC Biopharma. And that's a technology we call WaferiX. It's a sublingual wafer technology, and it's patented, it's novel. It's not invasive, it's sublingual, so you place it under the tongue. And it's been developed and manufactured by our CGMP facility in Australia, in Melbourne. It's a validated technology. We've now registered a pharmaceutical drug in Australia using this technology and have a number of others in phase 3, entering into phase 3 in the United States and pending approval elsewhere. So it's a versatile technology across a different number of pharmaceuticals, including cannabinoids, as I'll discuss in a moment. Importantly, it's quite a unique, it's manufactured in a unique way through a proprietary freeze drying process. The bulk suspensions in an aqueous or liquid form is filled into our plastic blisters that are formed in the manufacturing line. These are then go through a freeze dryer, which is a very complicated process over, it could be anywhere between 24 to even 36, 40 hours, depending on the product. And then they are sealed in that primary packaging to protect the wafer product. And the WaferiX technology is designed to do two things. Firstly, two things that you need to do to effectively administer a product sublingually. You need to disintegrate rapidly, and you need to release the active effectively. And it can do this because of the highly porous microstructure and the homogeneous dispersal of the active. And importantly, the actual wafer forms an amorphous matrix. There's a scanning electron micrograph picture on the bottom right there showing the irregular holes that at a microscopic level are formed. And within those irregular holes, the active drugs, CBD, THC, or both, can be contained. And because it's amorphous, it's a weaker structure that disintegrates and rapidly breaks down, releasing the active. The matrix is non-ionic, meaning it's non-charged and doesn't retard or hang on to the active molecules, CBD or THC in this case, or otherwise, and limit absorption across the sublingual membrane. The matrix is designed with a selection of forming agents, so to maximize disintegration, minimize dissolution time, and we use super disintegrants to aid with that, and also designed to maximize the transfer rate of the biological active across the sublingual mucosa, optimize the contact time between the drug and the mucosa. And all in all, this helps to minimize the swallowing of biological actives, which can be an issue. The limitation at the moment is in, I guess, the amount of drug that we can load onto our wafer matrix, but we have gone up to 100 milligrams, and for the purposes of most cannabis indications and a number of other pharmaceutical indications, this hasn't been a problem. In addition, multiple wafers can be administered at one time or one after the other. Of course, with sublingual delivery, as mentioned, the size of the molecule can be a limitation as well. So compared to other products that have been positioned for sublingual, particularly the solid dosage form ones like oral dispersible tablets or sublingual films, as they would call it, or film technology that's been suggested to place sublingually. The WaferiX has been specifically designed for sublingual administration, as opposed to repurposing from orally ingested products for nil swallow, no water, but they disintegrate and then they are ingested. That's quite a big difference. The rapid disintegration and maximal absorption allows absorption to be 2 to 4 times greater for oral than oral absorption with WaferiX, and that's by bypassing the GI and the first pass effect, and with the amorphous and non-ionic matrix. So there's then potential for faster and more predictable therapeutic action. It's a versatile platform, as I've mentioned, across multiple actors, and we've demonstrated 2 to 4 times improvements in oral bioavailability over oral absorption for those actives that are poorly absorbed, just like the cannabinoids, which are highly fat soluble. And these studies were absolute bioavailability studies in humans and have been published in highly regarded peer-reviewed journals. So now moving to CBD, well, we know that WaferiX can deliver CBD effectively using the sublingual approach. And on the right there, you'll see some pharmacokinetic data showing a 25 milligram CBD to 50 milligram CBD compared to 25 milligrams of the Oromucosal spray Sativex. So we see rapid absorption, we see less variable absorption, we see dose proportionality, which is important in dosing. So 25 doubling to 50 led to approximately a doubling of the maximum concentration and the total area under the curve, or total absorption. It was safe and well tolerated. And compared to Sativex, there was increased bioavailability for like dosing, increased peak plasma concentration, or Cmax, and a shorter time to peak plasma concentration, or Tmax. We then took the wafer and we took a nanoemulsion of CBD to combine the nanotechnology with our unique novel delivery system. And we saw further improvements in absorption, even more rapid absorption, higher Tmax, and greater bioavailability than what we'd seen previously with even our first generation WaferiX formulation with CBD, which was very encouraging. And so that product, now we've developed a product, Zativa, which was mentioned earlier, which is a sublingual broad spectrum nanoemulsion CBD product, and there are plans afoot for to combine CBD and THC and THC-only wafers, which we aim to have available later in the year. In terms of, just summarize on WaferiX, the advantages are they disintegrate within a minute, there's fast absorption, with the potential rapid onset of action, improved bioavailability, improved absorption, which given at the current situation in Australia where it's out-of-pocket expense for patients, private script, reducing the amount of cannabinoids required to treat a condition could have an economic benefit, and then more predictable absorption. Also fixed unit dosing, where you know exactly, and the prescriber and the patient knows exactly what they are being delivered. A long shelf life has been established with WaferiX as a technology. It's stable at room temperature for 3+ years. And with respect to the cannabinoid product that we currently have, we have a 12-month stability on that, a shelf life on that product. It's convenient, discreet, and very simple and easy to use. And on the right-hand side there, I'll just show you just a short video of what the wafer looks like and how it rapidly disperses in an aqueous environment. So when you place it under the tongue, it will break down within a minute. So with that, thank you very much, and look forward to speaking with you all at another time.